Okay, this is the last slide. So, did you like it? Okay. Well, I'm very happy to share part of my work today with you guys. I'm going to talk about how we try to understand brain inflammation. And I'm working on brain stroke. So what's brain stroke? Brain stroke is we know that there's two types of strokes. One is when you have a bleeding, so basically a blood vessel in your brain breaks, and there's a leakage of blood, and you have a hemorrhage. And the one that I study is the ischemic stroke. Ischemic stroke is similarly as what happens in your heart when you have a heart attack. A blood vessel gets obstructed by a blood clot, and your neurons and your cells in that region, a specific region of the brain, start dying because they stop receiving their food, which is basically sugar and oxygen. When this starts happening, you have inflammation occurring, which is what happens when you bruise an arm or anything, when you have an injury. So the tissue starts getting swelled. And why is this important? Because we have seen, or it's known, that inflammation can be bad or good at certain point. And what I'm interested about is which cells are responsible for brain inflammation. So we know that, and I thank Andy for the introduction because I'm talking about microglia and macrophages. So these are the two cells that are mainly working in inflammation in the brain, and these cells are very similar, as Sandy said. So microglia is part of the defense system in our body. Macrophages is also part of the defense system in our body. But microglia are restricted to the central nervous system, so we're going to find microglia only in the brain and the spinal cord, while macrophages are traveling through our body. So they're found in the blood, spleen, bone marrow, and they're like also searching for injury or tissues to infiltrate, because they have the property to infiltrate tissues after injury, like when there's a stroke in the brain. So my question is, do these cells, microglia and macrophages, behave differently after a stroke, or do they play a different role in inflammation after a stroke? And you might think, well, they're two different cells, of course they behave different. But before, we weren't able to differentiate between these two cells. So when we studied stroke, for example, a few years ago, these cells express the similar molecules. So we were talking about microglia and macrophages. And if you search articles, you always saw microglia or macrophages. Now, with genetic tools, as Andy said, we can put some colors into macrophages or microglia and differentiate between the two of them. And now we can talk about microglia and macrophages. And why is this important? Because this will give us a tool to study both cells separately and determine the role in different diseases of the central nervous system. In this case, a stroke. So what I do is I generate a stroke in a mouse. I take out the cells by a color. I stain them with different colors, and I separated both cells, and I study the gene expression, so the amount of DNA that these cells express. So I put these cells in a thermal cycler, which is a machine that will tell me what's the amount of a specific DNA or gene in these cells, and I focus my study in genes, in inflammatory genes. So genes involved in inflammation. And what I found is that there are specific genes involved in the inflammatory processes that are predominantly expressed or seen in the microglia, and there's some genes that are predominant in the macrophages. And this is important because at some point we might hopefully modulate the response or how these cells react to stroke, and hopefully we can treat the disease at some point. Thank you very much.